Cells are the smallest living units of an organism. Cells differ in shapes, structures, and functions. All cells consist of two main structures, no matter what type of cells they are. The first structure is cell membrane, which separates the inside of the cell from its environment. The second structure is protoplasm, which contains cytoplasm and organelles. In this lab session, you will observe the simple structures of some animal and plant cells that can be seen under a compound microscope. You will also study some functions of cells. Before starting to observe the cell structures under a microscope, a wet mouth slide should be prepared. This is an example of how to prepare a wet mouth slide for the epithelial cells from the surface of our oral cavity. Briefly, the epithelial cells can be taken by scratching inside our oral cavity with a toothpick. This step should be performed gently. Then, smear the epithelial cells on a drop of 0.85% sodium chloride solution on the slide. Finally, dye the epithelial cells with methylene blue solution and cover them with a cover glass. Other samples will be prepared using the same procedure. These are what we observe. Students may pause this video and draw pictures of cells in your report. Please label the organelles you draw. After the students observed the pictures of cells, let's discuss these questions. The second topic is the functions of a cell. Diffusion is the movement of a substance from a high concentration to a low concentration. The diffusion process can occur in both the liquid and gas phases. It is an important process for living things as it controls how substances move in and out of cells. In this experiment, the diffusion of ammonium hydroxide and hydrogen chloride was used as a model. This equation shows that ammonium hydroxide reacts with hydrogen chloride to form ammonium chloride, which is a white aqueous compound. The white ring of ammonium chloride will occur where both gases meet. These are the equipment and chemical reagents we used. This picture shows the setup of the gas diffusion experiment. Notice the white ring in the tube as shown in this picture. The distances from the cotton ball to the white ring from both sides were 440 millimeters and 160 millimeters. The reaction took 3 minutes and 52 seconds. Students should record this data in the report. The diffusion rate of each gas can be calculated from this equation. Please answer the 2 questions at the bottom of this page. The next term is osmosis. It is the movement of solvent molecules across a semi-permeable membrane from a region with a lower concentration of solute to a region with a high concentration of solute. Oyster plants' epidermal cells were used for demonstration. First, the epidermal cells were prepared in distilled water. Second, put a drop of 5% sodium chloride solution on one side of the cover glass, and the other side uses tissue paper to absorb the water from the slide. Sodium chloride solution will diffuse under the cover glass and will replace the water. Last, let the cell stand in the sodium chloride solution for 5 minutes, and then put a drop of distilled water on the side of the cover glass and remove the sodium chloride solution from the side using a tissue paper. Let's compare the changing of cells and answer this question. This is the last experiment. It is a study of selective permeability properties of the yeast plasma membrane. There are 7 tubes that contain the substances as shown in this table. In this case, neutral rate was used as a pH indicator. It turns red or pink in acidic solutions and yellow in alkaline solutions. Students will see the color change in this video. These pictures show the results in each step. The results of tube number 2 and 3 in step 3 were filtered and boiled respectively. Students may pause the video and record the color and discuss these questions.